Ribonucleic acid, or RNA, is a crucial molecule in genetics. There are three main types of RNA that work together to convert genetic information from DNA into proteins. These are messenger RNA, transfer RNA, and ribosomal RNA. Each type has a unique structure and plays a specific role in protein synthesis. Let's look at the three main types of RNA. Messenger RNA, abbreviated as mRNA, carries genetic messages. Transfer RNA, or tRNA, transports amino acids, and ribosomal RNA, known as rRNA, forms the protein-making machinery. Understanding these three types is essential to grasping how cells produce proteins. Messenger RNA acts as a molecular messenger. It carries genetic information from DNA in the nucleus to the ribosomes in the cytoplasm. mRNA is transcribed directly from DNA and contains sequences called codons. Each codon is a 3-nucleotide sequence that specifies a particular amino acid. Unlike DNA, mRNA is temporary and degrades quickly after use, allowing cells to regulate protein production efficiently. The journey from DNA to protein involves several steps. First, DNA is transcribed into messenger RNA in the nucleus. This mRNA then travels to a ribosome, which is the cellular structure where translation occurs. During translation, the ribosome reads the mRNA code and assembles amino acids into a protein chain. This process is fundamental to all life. Transfer RNA serves as the delivery system for amino acids. Each tRNA molecule has two important regions, an anticodon that recognizes specific mRNA codons and an attachment site that carries the corresponding amino acid. The unique cloverleaf-shaped structure of tRNA allows it to perform this dual function. By matching anticodons with codons, tRNA ensures that amino acids are added in the correct sequence during protein synthesis. Here's how tRNA works in practice. The anticodon on tRNA, such as USC, pairs with a complementary codon on mRNA. Like a UG, this base pairing follows strict rules where uracil pairs with adenine and cytosine pairs with guanine. When the match is correct, tRNA delivers its attached amino acid to the growing protein chain. For example, the codon AUG codes for the amino acid methionine, which is often the starting amino acid in proteins. Ribosomal RNA is the most abundant type of RNA in cells, making up about 80% of total RNA. rRNA combines with proteins to form ribosomes, the cellular machines that build proteins. But rRNA is not just structural. It also has catalytic activity, meaning it directly catalyzes the formation of peptide bonds between amino acids. This makes rRNA a ribozyme, an RNA molecule with enzymatic properties. Let's compare these three RNA types. mRNA carries the genetic code from nucleus to ribosome and is short-lived, comprising only 3 to 5% of cellular RNA. RNA delivers amino acids in the cytoplasm, is stable, and makes up 10 to 15% of total RNA. rRNA forms the ribosome structure, is very stable, and constitutes about 80% of all RNA in the cell. Together, these three types orchestrate the complex process of translating genetic information into functional proteins.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.